Good afternoon, everyone. So my talk today is on the immunogenicity profiling of pharmaceutical nanocarriers. The main focus of my talk today is on the immunogenicity profiling of pharmaceutical nanocarriers is on the cytokines and nanocarriers. However, the same exact principles can be also applied to other tested materials and to other biomarkers. So before I start, I'd like to thank Luminex and Biorad for this kind invitation. I have been in several conferences, but this is the first time in my life to be exposed to that intensive amount of information about the xMAP technology, because usually in different conferences you will see like a couple of slides on like using the technology, but this time most of the questions that I was looking for and several answers and experience I have seen during the very interesting talks. So the contents of my talk today will include some basic concepts on the nanomedicine, the use of cytokines as biomarkers for nanomaterials immunotoxicity, and the use of the immunotoxicity index as an easier way for data mining and interpretation, and some important consideration for the precise and accurate use of the xMAP technology. And then I'll give some few examples on the successful use of this immunotoxicity index in evaluating the immunotoxicity of nanomaterials. And finally, a short suggested plan to improve the science beyond the xMAP technology and broaden their application area, and to improve the scientific outcomes for the users. So my main interest is in the drug discovery and development, since I have started my undergraduate pharmacy studies and then my master and PhD degrees in pharmaceutical technology in University of Montreal. We have been using several types of drugs, ranging from small antimicrobial chemotherapeutic molecules to relatively larger nucleic acids and proteins, embedded into different types of nanocarriers, lipid-based or polymer-based, spherical or elongated, for either intravenous or topical or oral administration. We have been using several in vitro and in vivo characterization techniques to study the physicochemical and biological characteristics of these developed materials. And we have developed several systems for treatment of different diseases like diabetes, irritable bowel syndrome, hyperlipidemia, cancer, viral infections, or to overcome drug incompatibilities, and also for some topical antimicrobial and hemostatic applications. As we all know, there is a long journey for any new developed drug or delivery system from discovery to the market, and safety remains as a crucial part of this journey. Immunogenicity constitutes an important part, and cytokines can provide important information on the immunotoxicity of nanomaterials. Recently, we have found also they can be used to partially predict the in vivo pharmacokinetics and biodistribution of these materials. And this actually can save a lot of time and cost and also risk before testing these materials in vivo, either in animals or in humans. For more information on the use of cytokines as biomarkers for nanoparticle immunotoxicity, you can be referred to this review published by our group. Multiplexing technique, as we all know, include incubation of samples, either tissue or cell culture, supernatants, lysates, or serum or plasma that contain the pro-inflammatory mediators with multiple coded beads to allow the formation of the antibody detection complexes. And I know most of the people here are familiar with the technique and the different steps. So what I'd like to talk about first is challenges towards the precise and accurate use of the xMAP technology. And several of them have been covered in the first two talks, and it was very nice to explain the details of these challenges. However, my main focus today will be on the accuracy and data analysis, because the cost can be compensated for with the intensive amount of data generated, and for accuracy and handling, they can be much improved if we provide the users with the appropriate guidelines. So what I'm mainly concerned about is the accuracy and data analysis. So for accuracy, there is a potential interaction between the tested materials and biomarkers. And although it had been reported previously in literature, it has been also overlooked. So people know and several authors reported the potential interaction between tested materials and biomarkers, but most of the studies, whenever they perform the assay, they neglect this part, and they just measure the expression of different cytokines. However, this potential interaction might generate artifacts and lead to data misinterpretation due to reducing the available concentration of cytokines or other biomarkers available for the analysis. So here is just an example that the presence of one type of polymer, it's called polycarboxybutane. The presence of this polymer during the assay, it didn't influence the available concentration of interleukin-3 or interleukin-4, as we can see. However, when the same polymer is assembled in a form of nanoparticles, it reduced the available concentration of interleukin-4 as compared to the same exact concentration measured in the absence of the nanomaterials. The extent of this interaction depends on the nature of the cytokine and the physicochemical characteristics of the tested materials, like their size, charge, hydrophobicity, and many other characteristics. So the important point here is that it is very important to pre-assess the potential interaction between the tested materials and the biomarkers before running the assay. The second part is about the data analysis. Significant advancement in multiple analyte profiling has been achieved thanks to the technological breakthroughs in the polychromatic flow cytometry and multiplexing technique, which could generate intensive amount of data. However, comparing hundreds of biomarkers is tedious and impractical, especially now where we can assay up to 500 biomarkers in one well. And although this problem is very well recognized, a little has been done towards appropriate data management and analysis. So comparing these hundreds of biomarkers, either by the number of induced cytokines or biomarkers or by their level, is very tedious. And at the end, especially, okay, I know most of the speakers, as I have seen, they are kind of like expert in immunology or multiplexing assay. But what I would like to say are many other labs either doing some drug delivery or some development of pharmaceutical or medical devices. They are not expert either in immunology or in multiplexing technique. But it's very important for them to know the induction of these biomarkers by their materials, because this is like directly influence the safety of their tested materials. So for them, when they have these thousands of numbers, it's inconclusive for some of them and time consuming for others. And so at the end, and this is what's happening now, like most of the users are experts in either immunology or in the multiplexing technique. But if we provide them with solution for simple data analysis and processing, this can improve the science beyond the xMAP technology, broaden their application, and of course also will increase the sale of these kits. So just very brief concept on nanomedicine, because I'll give some few examples of them. So when we say nanomedicine, it means that we are designing the therapeutic and or diagnostic agents on the nanoscale for coincident transport and delivery of the active species and to control their navigation within the biological system for treatment, prevention, or diagnosis of the diseases. There are several types of nanomaterials with different structures, but most of them would share a core to load your cargo or your drug inside or your diagnostic agent, and then a shell to improve the stability of the nanocarrier and to mask the content of the core from the immune system. And then you can label these nanomaterials with different radio labels, with fluorescent group, with some targeting ligands, such as antibodies or peptides, to allow the recognition by specific receptors in the body. They can be also cross-linked to further improve the stability. For more information on the design of like these pharmaceutical nanocarriers, you can be referred to this review, or also for the combined use of diagnostic and therapeutic agents for drug delivery applications. So there are many factors to be considered in the design of pharmaceutical nanocarriers, from the preparation to the therapeutic response. Because it is beyond the focus of this talk, I just would like to highlight the main goal of our research is to convert the traditional therapy, where you give the drug and the drug being distributed everywhere in the body, in the different organs, it will go to the organ of interest to treat the disease, and again to the other body tissues, which destroy these tissues and cause a lot of adverse reactions. So we would like to convert this traditional therapy into targeted therapy. So in this way, we concentrate most of the drug or most of the dose into the liver, for example, if we are treating liver cancer. And in this way, we can increase the therapeutic efficiency, reduce the adverse reaction, and the dose and the cost associated with the medication. So nanomaterials, like any other exogenous materials, may unintentionally interact with the various components of the immune system. Even when you do like in vitro tissue culture assay or even in vivo, and there is no apparent toxicity, they may still trigger the immune system via several mechanisms and mediators. And this may lead to adverse reaction. This most of the researchers know, but also they can terminate the therapy or reduce the therapeutic efficiency associated with the medication. And this many people don't know. So it is not just inducing adverse reaction, but the therapeutic efficacy of the drug itself in the pharmaceutical carrier, the therapeutic effect can be reduced in case of induction of high levels of cytokines. Of course, the extent of this immune response depends on the physicochemical characteristics of the nanocarriers, and also on the drug payload and route of administration. The route of administration directly influences the immunomodulatory effect of these nanomaterials, because it will determine the type of the immune cells that will interact with these tested materials. So if you inject them and they will be directly in the blood interacting with the different immune cells or plasma proteins, it is different when you apply them topically or when you have them like dosed in capsules or in tablet orally. Because as we all know, there are different types of immune cells distributed in the different body organs or in the blood. One more factor also is the intracellular trafficking pathway. So the type of the receptors or the type of the cells that they are interacting with also affect the immune response to these nanomaterials. Cytokines. Cytokines, as you all know, are proteins that perform pleiotropic functions and play an important role in regulating the immune system. And in particular, the pro-inflammatory cytokines serve as mediators for inflammatory and immunological reactions. So if we can monitor the expression of these cytokines together with other biomarkers, we may be able to control the undesirable effect from the tested materials. And as I have mentioned, we have recently found, and I'll explain it later, that they may predict some of the in vivo performance of these tested materials, which can be very useful in predicting these parameters before testing these materials in vivo in animals or humans. Unfortunately, the lack of adequate data mining and management techniques limits the ability of several users of optimally using the generated data, and also necessitates the development of analytical tools that can produce descriptive values for each tested materials, and these values should be simple enough for non-experts to use. So the examples that I usually give, I don't know if anyone here is familiar with the cytotoxicity assays, where we incubate the cells with different concentrations of the tested materials and measure the cell viability. If you have these two sigmoidal curves and you're trying to compare point by point at different concentrations, it will be almost inconclusive, because you can say at 5 microgram per milliliters, like this material A is more toxic than B. And then at another concentration, this toxicity pattern may change. So it's very confusing. That's why we have usually the IC50 value, which is a value at which the tested materials kill 50% of the cells. So we use single descriptive value for cytotoxicity to describe the toxicity of the materials in vitro. Same thing in vivo, when you do like in vivo toxicity with the LD50 value, or for pharmacokinetics, where you have the half-life, the clearance, the area under the plasma concentration time curve. For biodistribution, where you have the percentage of the dose in different organs. For pharmacodynamic, where you measure particular response. And this is the same thing that we need for the immunotoxicity, or the expressions of these different biomarkers. You need something like what we call initially immunotoxicity index. So like single descriptive value, like all other these tests that can describe the immunotoxicity or the induction of different biomarkers by the tested materials. How we calculate, I'm not going to the much details of how to calculate it, but it is very simple. So we have like calculated numerical values by measuring the expression of the cytokines that had significantly higher expression than the control. When we say control, if for in vitro, for example, tissue culture, it can be the untreated cells, and in vivo, it can be the animals injected with saline or other controls that we have. And then we normalize the level of these cytokines to our control and take the sum of all these values and use it as the immunotoxicity index. And the higher the immunotoxicity index, the higher the immunotoxicity of the tested materials. However, there are some experimental conditions that must be defined, like the concentration of the materials and the sampling time in case of in vivo experiments, or the incubation time for in vitro experiments, because these values directly influence the magnitude of the immunotoxicity index. Using like this immunotoxicity index, just in one quick example, we could convert like the figures that you can see now is exactly 60 figures and 190 values, when the data were presented as like one figure for each tested cytokines between the different materials. And here it's not only like summarizing all this amount of data, it has exactly the same conclusion. Same conclusion could be attained with even more accuracy, and you can compare. So you can see from one graph like the values, like you can expect which materials will induce high expression of these cytokines as compared to other materials. Hint on the use of like this immunotoxicity index has been published by our group in Accounts of Chemical Research, but more details on like guideline for how to avoid the interaction between like the tested materials and the biomarkers, and on the use of like this immunotoxicity index. So hopefully it will be incorporated into kind of like software, will be developed hopefully in the future with like one of the companies that's using the xMAP technology. So now I will give some examples on the successful use of the immunotoxicity index to evaluate the immunotoxicity of nanomaterials. So when initially we performed these studies and published it, we didn't have the immunotoxicity index, and we spent several months for data analysis as many other groups. But after we came up with this value, like we could came up with the same exact conclusion with even more accuracy in just few hours. I'm not going in details beyond the scientific details about the design of these nanomaterials because of the time limit, and I will just highlight how the immunotoxicity index could replace these like huge amount of data by few descriptive values. So in the first example, we were synthesizing biodegradable polyphosphoester nanoparticles, and we studied the effect of surface charge, like when these nanoparticles have like a neutral, cationic, anionic, or zwitterionic charge on their surfaces, and how this can affect their toxicity. So we measured the toxicity of either the nanoparticles or their degradation products. Also, we examined the effect of cross-linking with mouse raw macrophages. And when we like initially performed this study, like most of them induced low secretion of cytokines as compared to lipofectamine, which is a commercially available transfection reagent for delivery of nucleic acids. At that time, when we had this huge amount of data, the only way that we could do it, we compared the number of induced cytokines. And then we could rank them as like the cationic nanoparticles were the most toxic, which is usually, as reported in literature, usually any particles or any delivery system with cationic surface charge will be more toxic than, like, for example, neutral or like anionic system. And then when we analyzed them based on the immunotoxicity index, we get exact same way of ranking between the highest toxic and lowest toxic nanoparticles, but in much faster way. And it was much easier than just counting or analyzing the different types of cytokines. Interestingly, also, we could produce quantitative data. So, for example, I mean, when you say lipofectamine, you have immunotoxicity index of around like 50. And for the most toxic materials that we have tested, it was around 20. So at least you have numbers to compare. I'm not saying like this immunotoxicity index should completely replace like the data that you generate. Of course, you need the different types of cytokines to understand why is this one being induced and like which ones are higher. But at least a value that you can use to explain your data, or at least for screening in the beginning before going to the details of your data. And this study was published in Scientific Report in collaboration with the Department of Chemical Engineering at MIT. In a second study, our main interest is to design multifunctional nanoparticles. So in this study, we have synthesized cylindrical nanoparticles with hydrophobic core. This hydrophobic core was used to load paclitaxel, which is an anti-cancer drug. And these cylinders have an ionic surface. On the anionic surface, we have templated cationic spherical nanoparticles complexed with short interfering RNA, and their surfaces are decorated with F3 tumor homing peptide. Then we had several characterization, physicochemical and biological, on the properties of these hierarchically assembled theranostic nanostructures. One of them was the immunotoxicity. And as I have mentioned before, we usually expect higher immunotoxicity for the cationic nanoparticles. And usually it is the lowest for anionic or neutral nanoparticles. Here we found even the anionic cylinders have high immunotoxicity. It induces the release of several cytokines. And the assembled nanostructures had the least immunotoxicity. I'm not going on the details about explaining why are these differences, but again, same situation when these data were published, we had to explain them with tens of figures either in the manuscripts or in the supporting information. But when we use the immunotoxicity index, we could generate everything in one graph in few numbers, and it was exactly the same conclusion from the paper. In this study, we have loaded biodegradable nanoparticles with cisplatin, which is an anti-cancer drug, and studied the effect of cross-linking on the immunotoxicity of the nanoparticles. And in this case, the difference between the two types of nanoparticles was small. So I mean, when you compare every cytokine, the difference was really small. So at that time, when we compared the number of induced cytokines, we got to the conclusion that the nanoparticles with higher degree of cross-linking have more immunotoxic effect. Although this was contrary to what we have observed before and what people have observed before. But then when we applied the immunotoxicity index, because the difference here was small, so the immunotoxicity index for the nanoparticles with higher cross-linking degree had lower immunotoxicity. Because when you cross-link the nanoparticles, you prevent them more from interacting with the immune cells. So here, in this case, even it's not only easier to use the immunotoxicity index, but it was also more accurate when the difference between the tested biomarkers are small. In this example, we have synthesized polyacrylic acid, polylactic acid copolymer, and then we functionalized them with different functional groups. And the main focus here was to examine the effect of polymeric coating. So when we coat these nanoparticles with polyethylene glycol, which is the most commonly shell-used polymer for most types of nanoparticles and pharmaceuticals, versus polycarboxybutane, which is the viterionic polymer. So we wanted to study the effect of this polymeric coating on pharmacokinetics, biodistribution, and immunotoxicity of these nanoparticles. So the uncoated nanoparticles, the particles without polymer coat, induced high amount of cytokines released from the tested cells, either in vitro or in vivo. The polycarboxybutane coated polymers induced the release of higher amount of cytokines. The nanoparticles that had the higher in vitro immunotoxicity were correlated with longer blood circulation and lower clearance in the immune organ. Again, also in vivo, the PCP-coated nanoparticles resulted in secretion of higher amount of the tested cytokines. And also the same nanoparticles induced the absorption of different proteins to greater extent than the polyethylene glycol. So just to summarize from this study, I know it might be confusing because I don't have time to go to the details of the study. What I want to say here is when we tested the in vitro immunotoxicity, just how are these nanoparticles are inducing the release of different cytokines. We found that the nanoparticles associated with release of higher amount of cytokines in vitro have the same pattern in vivo. And they also absorb more proteins on their surfaces, which is associated with more opsonization in vivo, and then more clearance in the immune organ and shorter circulation time. So we have kind of correlation between their ability to absorb proteins or opsonins, and their ability to induce the cytokines in vitro and in vivo. So here we may be able to use them as predictor for the in vivo pharmacokinetics and biodistribution. But of course, we need much more studies to confirm that these parameters can be used as predictor for the in vivo characteristics of these nanoparticles. Then this will be the last example where we studied the effect of cross-linking on the immunotoxicity of nanomaterials. And the idea here is that you have these nanomaterials or nanoparticles assembled from different polymers. When you leave them without any cross-linking, they can then dissociate and interact with the different types of immune cells. But when you cross-link them and limit the release or the dissociation of the free polymers, you reduce the interaction with the immune cells and reduce the immunotoxicity of these nanomaterials. When we tested their immunotoxicity, we examined the effect of different polymers, we examined the effect of different doses, and what we get is hundreds of numbers. And we spent several months of analysis, and even in the discussion of the paper at that time, we presented tens of these figures, and it was very tedious. So we go to every cytokine, and okay, for this particular cytokine, these nanoparticles induce the release of higher amount of cytokines, and then by increasing the dose, this is what happened, but the pattern was different for the second cytokine, so it was very tedious to go back and forth between the different cytokines. And after we published this paper, and we applied the immunotoxicity index, oh, sorry, this one doesn't work. It doesn't work. Okay. So then we could replace all this amount of data with only this figure. And as you can see, you can get everything. For lipofectamine, which is a commercially available transfection reagent and known to have high immunotoxic effect, you can see the immunotoxicity index. And then you can compare, if you can see, here, by increasing the degree of cross-linking, you have lower induction of the tested cytokines, either at 5 or at 50 microgram per milliliter. And here, for example, you can see that for this type of nanoparticles, it induced the release almost half quantities of the cytokines as compared to the lipofectamine, although the dose here was 10 times the dose of the used lipofectamine. So again, I think in the future, we should use these kind of values at least to give description of the immunotoxic effect or the release of the biomarkers in terms of quantitative data. And still, you can take any kind of cytokines, or you can look at the detailed data. And if you are interested to understand the mechanism beyond this immunotoxic effect, but the use of this descriptive value will help in simpler data mining and analysis. So in conclusion, we have used these values to publish several papers and to understand the relationship between the structure and composition of nanomaterials to the immunotoxic effect. We could give some recommendations, on which type of coating, of polymeric coatings that you should use, which polymers that will have lower immunomodulatory effect, which type of cargoes, drugs or nucleic acid, can induce higher immune response. What about the targeting ligand, what it can do with the immune system. For the shell of these materials, what is the effect of cross-linking? We could draw several conclusions, and the same thing can be applied for other pharmaceutical or medical devices. So immunotoxicity index provide descriptive value for each tested materials. And we expect, with this immunotoxicity index and the use of appropriate guidelines for precise and accurate measurements of biomarkers, that can provide very useful information for the users and may also help in predicting their in vivo pharmacokinetics and biodistribution. At the end, I just have also suggested plan for the companies who are using the xMAP technology, and to improve the science beyond the xMAP technology and broaden the application area and improve the scientific outcomes for the users. Just to improve, they are doing great, but with people who are expert in this field. But for many other peoples in several, like pharmaceutical nanotechnology or drug delivery, and many other labs or pharmaceutical industry who are not expert in the measurement of different cytokines, they need like more information, because when you just provide them with the kits, they can do it, but after they are generating thousands of data, they don't know what to do. I mean, they don't know how to analyze them, how to understand them. So if we increase awareness to them on how to use this technology for simpler way for data analysis and mining, so this will provide them with much easier way of acquiring data that will be useful for them for design of safe biopharmaceutical agents and medical devices. So I'd like to acknowledge Professor Karen Woolley, the director of the LSPI at Texas A&M University, and also her group for providing these beautiful nanostructures, which allowed me to understand the relationship between the composition and the different parts of the nanomaterials and the release of the different cytokines in response to these materials. Several also of these projects were supported by the NIH, NSF, and the National Heart, Lung, and Blood Institute, and National Institute of Diabetes and Digestive and Kidney Diseases. So thanks. And this is just my message. Yeah, thank you.